I have the pleasure of introducing our seminar speaker today, Dr. Bobbi Langkamp-Henken. She worked for 6 years as a hospital dietitian before pursuing a doctorate in gastrointestinal physiology, a postdoctoral fellowship in immunology at the University of Tennessee Health Science Center. Currently, Dr. Langkamp-Henken is a professor in food science and human nutrition department at the University of Florida. Our main research interest is to develop novel approaches to examine the effect of dietary interventions on health outcomes in healthy children and adults. Ongoing interventional studies explore the impact of whole foods, fermentable fibers, and probiotics on GI and immunological functions important for maintaining health and quality of life. Dr. Langkamp-Henken also teaches 2-semester undergraduate research planning courses and serves as graduate coordinator for the doctoral program in nutrition sciences at the University of Florida. So we'll all welcome Dr. Langkamp-Henken for coming and talking with us today. And I'm so excited to hear your research.
All right, well, let me share my screen. Okay, and let's see. It's responding a little slowly. All right, are you seeing just the slides now? All right, very good. I'm going to move all your pictures a little bit. All right, so again, thanks for the nice introduction and for the invite to speak with you today. As way of disclosures, I have a list of the current funding sources that I have right now, as well as the funding sources that were used to produce the research I'm going to share with you today. As was stated, my main research interest is to develop models to examine the effect of interventions such as pre and probiotics and fiber on gastrointestinal function and health outcomes in healthy individuals. So what I'd like to do today is discuss why would you want to look at health outcomes in healthy individuals, and then what are the challenges, and there are many, to finding measurable differences in participants when they're healthy as opposed to ill. And then what are the novel models that we can use to increase the magnitude or frequency of outcomes to find differences between intervention groups. So just a couple definitions before we get started. I don't know if you're aware or not, but in 2017, the International Scientific Association for Probiotics and Prebiotics wrote a consensus statement where they kind of modified the definition of a prebiotic. The definition currently is a substrate that is selectively utilized by host microorganisms, conferring a health benefit. And it used to be a carbohydrate. It's no longer limited to carbohydrates. The prebiotic can be natural or synthesized. Examples would be inulin, fructooligosaccharides, galactooligosaccharides, and human milk oligosaccharides. But the definition allows for conjugated lipids and polyphenolic compounds to be called prebiotics at some point. The health benefit has to be shown first for these compounds before they can be termed official prebiotics, but I'm sure the research is coming, and I'm trying to do some of that research as well. For probiotics, the definition is live microorganisms that, when administered in adequate amounts, confer a health benefit to the host. And examples would be Lactobacillus gasseri, KS13, or Bifidobacterium infantis, R0033. And the reason I'm giving you genus, species, and strain is that if we do studies, we need to make sure that we list the genus, species, and strain, because different strains can have different effects on health outcomes. All right, so why study the effect of pre and probiotics in healthy individuals? Well, for one, the FDA states that dietary supplements are intended only to affect the structure or function of the body and not intended for therapeutic purposes. So in other words, if you use a probiotic or a prebiotic to diagnose, cure, mitigate, treat, or prevent a disease, your probiotic or prebiotic now becomes a drug. And as a drug, it can be regulated such that you would need a prescription to buy this prebiotic or probiotic. Dietary supplement companies don't want that to happen. They don't want their prebiotic or probiotic to be classified as a drug, because then they won't sell as much. They'll sell more when it's on the drugstore or the grocery store shelf and you pull it off, as opposed to you having to get a prescription from your physician to purchase that prebiotic or the probiotic. To give you an example of this, early in my career at the University of Florida, Dr. Wendy Dahl and I proposed the study where we were going to enroll students who came to student health for some type of infection, urinary tract infection or strep throat or whatever, and they were prescribed antibiotics. And we wanted to randomize them to receive a placebo or a probiotic simultaneously with their antibiotic. And so when we proposed this and we sent it into our institutional review board, they said, you got to talk to FDA first because we think this is a drug study. So we had to write a proposal to the FDA and they evaluated it. And they said, yes, I'm sorry, antibiotic associated diarrhea is a disease. And you can do this study, but you have to fill out an investigational new drug application and then do phase 1, phase 2, phase 3 clinical trials. Well, that takes a lot of money and that takes a lot of time. And so we just opted to pull our study instead of trying to do that study. But you can see how even in research as well as marketing, you have to follow the same rules. In research, I can't use a prebiotic or probiotic to cure, mitigate, or treat or prevent a disease, or it becomes a drug, and then I have to do the investigational new drug route. All right, so, but why would we want to study gastrointestinal symptoms and use prebiotics or probiotics? Well, because a lot of Americans have problems with GI symptoms. In a survey of 71,000 Americans, 61% reported one or more GI symptom in the past week. And when they asked these individuals who were surveyed whether or not they were healthy, 52,000 said they were healthy, but they still, 54% still reported one or more GI symptom in the past week, with higher prevalence in these groups. And the most common symptom being heartburn, reflux, and in descending order, abdominal pain, bloating, diarrhea, constipation. So generally healthy Americans have real GI issues and they're looking for relief. As a matter of fact, they spend $43 billion annually on supplements just to improve GI health, with very little data out there to show whether or not there's actually benefit of these supplements in terms of GI health in healthy adults. So that's why I study healthy adults. But it's really, really challenging to find differences in health outcomes when people are healthy to begin with. So what I have developed are different models in which the frequency and the magnitude of the symptoms are increased. So I'm better able to find differences between my intervention and my placebo. The other thing that's a challenge is that dietary interventions have very subtle effects. They're not drugs. Drugs have big time effects. Dietary supplements don't. So automatically the effects that you're going to be looking at in these healthy populations are much lower in magnitude or challenging to find. And to give you an example of that, I know some probiotics that might decrease diarrhea symptoms, and they work for some people, maybe not all, but if I were doing a study with a probiotic versus Imodium, I would bet that I could decrease diarrhea symptoms with Imodium, the drug, much better than I could with the probiotic. So the effects are very subtle. It takes a large number of people to see these measurable effects. And then on top of that, we're all different. And so there's a lot of inherent variation between you and me and everyone else. And so sometimes the variation between the participants is greater than the effect that you're actually able to measure. So I have some tricks and some advice for how to decrease some of that variation as well. All right, so what are some times in our lives, healthy individuals, where we might not be so healthy? We might have more GI symptoms. We might have more higher incidence of cold and flu. What are these at-risk times? Well, we know that stressed individuals, students, caregivers are more likely to have GI issues as well as cold and flu symptoms. I also know that if I enroll people with a large waist circumference, I'm probably going to get a lot of people with a high cholesterol levels. So I might be able to use that probiotic and indirectly look at cholesterol levels, because if I were going to do that outright, I'd be treating a disease, because cholesterol is a surrogate for the disease of cardiovascular disease. Aged populations, as we get older, our immune function declines. And so that's a great population to look at. Can we reconstitute the immune system? Can we shift the immune system to protect them from cold and flu? Elite athletes training for a marathon or triathlon have high incidence of GI issues and cold and flu. That would be another population, healthy population, that you could look at health outcomes. I'll also discuss today a model of seasonal allergy and a model where we know people are going to have more gastrointestinal symptoms. The most important thing, and what I do when I'm looking at a probiotic or a prebiotic, is I match what is known about that with the model. So if I know it's going to influence immune function, I might be looking at allergy or cold and flu. If I know it's going to influence GI function, I might be looking, like right now what we're doing in our labs, we're looking at menstruation or other issues where exercise training will have increased GI symptoms. So you can't just pick a model, you have to know which model to pick based on the mechanism of action that you anticipate from your prebiotic or probiotic. So we frequently use the stress response as a model in my lab. And when we get stressed, we increase our epinephrine and norepinephrine, and this fight or flight hormone redistributes the blood flow from our GI tract to our muscles so we can run away from that dinosaur. It also alters gastrointestinal motility and gastrointestinal secretions. Well, those changes then will alter your microbial populations and could result in a loss of intestinal barrier function. And that loss of intestinal barrier function and altered microbial populations can then alter systemic immune function. We want to keep away from that dinosaur. Our stress response needs to keep us awake. And so if you have a chronic stress, then you could have higher levels of anxiety and insomnia that could lead to depression. So if you think your probiotic has an effect on depression, a stress model might be a great model to look at. And then also the stress response mobilizes immune cells. In case you get wounded, you need immune cells for wound healing. You also need it to fight infection. And so that can result in altered immune function as well. This has been known for quite a while that academic stress is associated with increased risk of cold and flu. And these studies were initially done in medical students undergoing their academic exams. And they showed that during the exam period, the medical students had decreased natural killer cell cytotoxicity, they had decreased lymphocyte proliferation, they had altered cytokine production, they had decreased salivary secretory IgA, which is the major immunoglobulin or antibody in our mucosal secretions, and they also had decreased fecal lactic acid bacteria levels. So we capitalized on this. We first examined whether the stress response was there with our undergraduate population. We have like 50,000 undergraduate students, so we can run large studies because I know I can get at least 600 students at one time to run a study. And so what we've shown a number of times is if you enroll students somewhere around October or November, they have a stress level of 3 on a scale of 0 to 10, being extremely stressed. And they kind of bounce around 3 to 4 as their stress level. Stress levels decrease when they go home for Thanksgiving. And then, oh my gosh, I got to go back to school and I have exams coming up. So now I get stressed. And then stress peaks during the first couple days of final exams, and then students start finishing their exams and going home, so their stress level decreases, and then over winter break they have a very low stress score. And so we wanted to know if we could, well, the other thing I should mention is when I was teaching each year, I would notice that around final exams or during final exams, when I was watching all the students to make sure there was no cheating going on, not that anyone at UF would ever cheat, but we'd have to sit there and watch them. And there was a lot of coughing and sniffing and sneezing and during the fall. And so I thought, oh, good, they are stressed and they're getting sick. So let me see if I can prevent that. So one of the first studies we did is we looked at the effect of galacto-oligosaccharide, which is a prebiotic, on gastrointestinal function and percentage of days of cold and flu in our academically stressed students. So we enrolled them 427 concurrently, so they all ran together over the same period of time. They were full-time undergraduate students. They had to have at least one final exam to be enrolled in the study. We randomized them to receive 0, 2.5, or 5 grams of GOS every day for 8 weeks. And then each day they did online questionnaires, which asked about their level of stress and their cold symptoms and the intensity of the cold symptoms. And then they did a weekly gastrointestinal symptom rating scale that asked about GI symptoms. And what we found is when we looked at the gastrointestinal symptom rating scale, diarrhea syndrome, which consists of a number of symptoms like loose stools, urgent need to defecate, and actual diarrhea, that those that received the 5 grams of GOS had the lowest score. So they had less issues with diarrhea and diarrhea syndrome. Same thing with constipation, the 5 grams of GOS, there was less constipation compared to the 0 gram placebo. When it came to abdominal pain and indigestion syndrome, the 2.5 grams of GOS was actually the better in decreasing these GI symptoms. And then 5 grams was actually better than the placebo. And the reason we think that the 2.5 was better in this case was because GOS is a fermentable fiber. And what we think happened is that those that were on the higher level of GOS, they had more gas and bloating, which they did, that's part of the indigestion syndrome score. And so the GOS actually contributed to some of the GI symptoms, resulting in an elevated score. So these data show that GOS can actually result in fewer GI symptoms during an academic stress. We also looked at cold and flu symptoms. So let me just set up this slide first. This is level of stress from 0, and remember it went all the way to 10, but I'm just showing you 0 to 6. The first thing I want you to notice is that the average symptom intensity score for cold symptoms. So there were 9 cold symptoms and they ranked them from 1 to 3. And so this is the average score. At 0 levels stress, 1, 2, 3. Notice how as stress increases, the cold and flu symptom intensity score increases. So we know that stress was definitely associated with cold and flu symptom intensity. When we look at the GOS versus placebo, with no stress, there were fewer symptoms with 2.5 and 5 grams of GOS versus the placebo. This continued until stress level of 2. And so this is where we're losing this effect of the 5 grams again. And so we think again that they were fermenting the GOS and they were a little bit more stressed. And so they were having other symptoms that maybe contributed to their stress and increased their cold and flu intensity. But we definitely could modulate the average symptom intensity score with this prebiotic. We tried it with the probiotics as well. This was another study that we did, and this shows that we got about the same curve that we did in the previous study, where this would be the peak during final exams, and then they go on winter break, and it's this average daily stress score decreases. And we used placebo. We had Lactobacillus helveticus, Bifidobacterium bifidum, and Bifidobacterium infantis. And what we saw with the infantis was actually a decrease in the stress response with that probiotic. And We think this is from the gut-brain axis, and this has been shown with another number of probiotics that you can actually decrease the stress response by giving a probiotic. So when we looked at the proportion of students reporting a day of cold, we also noticed that there was a decreased proportion with the B. infantis. We also saw a little bit decrease with the bifidum, with the B. bifidum as well. So we were able to, with a probiotic as well, decrease not only proportion of days of cold, but also with one of them, we decreased the level of stress. When we looked at the data from this study I just showed you with the 3 probiotics, we had an effect, a significant effect of sex. Females, for every level of stress, 0 to 10, versus males, 0 to 10. The proportion of participants reporting a cold for every level of stress, females reported, a greater proportion of females reported a day of cold or flu. So when you do these studies, one part of the variation that you can control is sex, added as a covariate in your model, so you can partition out the variation that comes from being female or male on some of these outcomes. We see this consistently when it comes to GI symptoms between sexes. Males tend to have more bowel movements. Females have a higher abdominal pain syndrome score and indigestion syndrome score. And when they self-report constipation, there's a greater proportion of females that report a day of constipation, or the proportion of days in a week with constipation, than what males do. So one of my undergraduate students thought that was very interesting, and she wanted to do a study to see if these differences between females and males could be accounted for by examining or looking at the menstrual cycle. And so what she did is she enrolled healthy young women taking oral contraceptives. And the reason we chose to enroll women who are on contraceptives is then they all, in theory, should have had a 28-day cycle, and we could match them up based on the day that they would start menstruating. And then we followed their gastrointestinal symptoms over the menstrual cycle. And what Taylor found was, this is day of menstruation. So first day of menstruation is day 1. This is day of ovulation. We're looking at mean daily GI scores by symptom. And this would be on a scale of 1 to 7. I'm just showing you 1 to 3 here, so you can see the differences between the different GI symptoms: abdominal pain, indigestion, diarrhea, constipation, and reflux. And what she showed is the first day of menstruation, all of these were higher than any other day in the menstrual cycle. So definitely some of the variation due to females probably is due to the menstrual cycle. Additionally, so that was with GI symptoms, she showed that stress also does the same thing. So this blue line represents self-reported stress on the scale of 1 to 10, 10 being the worst. Here's day 1. You can see the day before, during, and after, and leading up to it, women have a higher stress. So that could also play into some of the differences we see in our stress model. This represents Bristol stool scale score. So it's how formed the stool is. A score of 1 would be like rabbit pellets, and a score of 7 would be liquid. And so on the day of menstruation, women have softer stools. And this last one is stool frequency. So they have more stools too on that day. So Taylor now is one of my doctoral students. And so she proposed, and actually got this study funded herself. I'm so proud of her. She proposed to enroll healthy, regulating, menstruating women who typically take oral contraceptives and do experience GI symptoms. So we're kind of padding our, or making this a little bit more successful for us by enrolling women who say they typically have GI symptoms. So that'll increase the frequency of GI symptoms that we'll see, and we'll be better able to see differences between the probiotic and the placebo. So she's going to then follow them, give them the probiotic or placebo for a month, and then look at the effect on the menstrual cycle the following month. Her proposed mechanism is that the probiotic will shift the intestinal bacteria that is associated with health, normalizing GI function, and reducing the stress response. And I can't tell you about this right now. She just finished enrolling participants, and the last participants have not finished yet. And so you'll have to stay tuned for another year probably before you see the results of the study. But we're really excited that I'm excited for her too, that this was totally her idea. And she got it, as I said, she got it funded. Other models that we have currently going in our lab or we've used in the past, right now we are doing a study looking at non-elite runners. And the reason we chose to look at non-elite runners who are in training for events as opposed to elite runners is that we think elite runners have kind of maxed out any changes that we hope to see. So remember I told you probiotics, prebiotics have subtle effects. Well, they might just be totally overwhelmed in an elite runner. So we are enrolling non-elite runners who are in training, and we know from the literature that training and running a lot can result in GI symptoms and increased incidence of cold and flu, which then results in loss of training time. So our hypothesis is that, or we're proposing that the probiotic will actually allow them to train more. And so then we're looking at the effects of time to exhaustion in these runners to see if we can change that between baseline and our final measurement. It could also be that if we have more short chain fatty acids produced, I mean, that's a source of energy, maybe that'll spare some of the glucose and that might actually help too. So there are a lot of other reasons why this might work, but this was kind of the main one that we picked our probiotic based on the GI symptoms and cold and flu. We've used an aging model in my lab a number of times because we know immune function declines with age and older adults are more susceptible to cold and flu. And so we've given different fibers, prebiotics, and probiotics, and to see if we could modulate immune function and then health outcomes. So this is one of my really cool models that we've done recently. One of the aging studies that we did with the probiotic, we showed that the probiotic as a combination probiotic actually increased IL-10 concentrations in vitro. We got a little excited because this probiotic combination contained B bifidum G9-1, and so it increased the IL-10. This same probiotic had been used in mice, and it had been shown to decrease allergy-specific immunoglobulin E. And so if you have an allergy, you take in the allergen, your Th2 responses increase, you make immunoglobulin IgE, it binds to mast cells, they degranulate, and you get runny nose, itchy eyes, and increased nasal pressure. And so we knew we might be able to decrease IL-10. We knew from mouse studies that it might have an effect on IgE, and there were actually guinea pig studies that showed that this very same strain of probiotic decreased nasal pressure and sneezing in guinea pigs when exposed to an allergen. So we thought we'd test it in an allergy model, but we can't treat allergy, right? Because then it becomes a drug. So we had to get creative. And so instead we looked at quality of life. And from somebody who experiences a lot of seasonal allergies, improving my quality of life would be as important to know as how to use less labor or whatever. So Jennifer, in this study, she found a questionnaire, a quality of life questionnaire related to allergy, and it was the mini rhino-conjunctive quality of life questionnaire. We also did have secondary outcomes to try to find a mechanism, looking at serum IgE, the percentage of circulating Tregs, which are part of our tolerance. So it kind of dampens an allergic response. We also looked at fecal microbiota profiles and GI symptoms. Just so happens that a researcher at UF was collecting pollen on top of one of the buildings on campus for a number of years. And so we had a pretty good record over the years as to when the pollen really increased in Gainesville, Florida. And so we designed the study. Our goal was to kind of pre-feed participants either the probiotic or the placebo for about a month or so prior to peak pollen counts to see if then we could improve quality of life. And so we had 84 in each group, but we had a subgroup where we collected the blood and stool because we would need fewer to see differences in blood and stool. But since we're using a questionnaire, we needed a larger number in the big group. This was what the questionnaire looked like. It asked, over the past week, have you been troubled by these various, by nose and eye symptoms so that you couldn't do these activities, or that you needed to rub your nose and eyes all the time, or you needed to blow your nose, or you had sneezing or whatever. And so this is what the mini RQLQ looked like. What we found was with this probiotic in blue versus the placebo in orange, we were able to decrease nose score, so sneezing, stuffy nose, runny nose between final and baseline and final. And the reason I say decrease is because we had an atypical year and pollen counts were high actually at the beginning of our study. So we were actually looking for a decrease then. We thought symptoms would increase over time because we had, our final was at peak, but it didn't quite work out that. But we were able to show a decrease in nose, other, activity score, practical problem score. We did not see a change in the eye score. We did look at IgE and Tregs, and they were high in both groups, meaning that they were suffering from allergy. And we think, again, that it was elevated in both groups because it was elevated at the start, because we predicted the allergy season we were off by about a month. But we also showed fewer constipation symptoms, which to us meant that we were modulating gastrointestinal function. And when we looked at the fecal microbiota, OTUs corresponding to Lactobacillus gasseri increased, which was not a big surprise because that was actually one of the components of our combination probiotic. But we also showed an increase in OTUs related to Faecalibacterium, which is has been associated with health in many studies. We saw a decrease in prevalence of E. coli and parainfluenzae. So from what I've shown you right to this point, hopefully you got the idea that probiotics and prebiotics may offer some benefit during life stresses. The big question is, are they beneficial during other periods? Should I take a probiotic or a prebiotic supplement every day? Wow, that's a good question. I would like to study it. It would be hard to study. To do this, we would need probably a large sample size, very long study, because we'd have days where you weren't a high risk for whatever outcome, intermixed with periods when you were at higher risk for the outcome that we were looking at. And we have, as I mentioned earlier, a lot of individual and study related variation that we would need to decrease. So how can we decrease some of that variation to be able to measure changes that are pretty subtle when it comes to prebiotics and probiotics? One of the things you can do is tighten up the inclusion-exclusion criteria. Maybe, remember I showed you there's a big difference between males and females. Why not just enroll males? Don't even enroll females. Well, the push from NIH is to include everybody, all socioeconomic levels, all races, all ethnicities, etc. So I'm not sure that's a really good idea, but maybe we can control for them at the back end statistically. And the other thing too is if you start really tightening up your inclusion-exclusion criteria, then who is it applicable to? You know, it's only applicable to males with a BMI that's normal on a Wednesday, you know, in the odd month, you know, that it works. So every time you add a lot of inclusion-exclusion criteria to decrease variability, you're kind of changing your target population. Another way you can decrease variation is you can do a crossover design. So each participant serves as his own control. And industry right now that we are working with, they really are all 4 crossover studies? Well, it's because you need a smaller sample size because each person is his or her own control. But I vowed I will never do a crossover study again in my life because they're so hard to interpret. There's so many other things that can go wrong. You can have order effects, carryover effects, period effects, learning effects. You have increased risk of participants dropping out because your study is like really long now. So for the crossover, you randomize someone to treatment A, and they have a washout period, and then they get treatment B. And you'd have another group that started with B, washout, and then treatment A. And so this makes for a very long study. If I were doing a parallel design study, I would just have this part. I'd have a lot more sub participants, but it'd be a much shorter study. So I mentioned that we did an aged adult study, and this is the study that we did, and it was a crossover. So I'm going to show you what a mess it was to interpret when we got these results. My graduate student used this combination probiotic that we used in the allergy study. This study was actually done before the allergy study. And so it was Lactobacillus gasseri, the bifidum, and the longum. And Sam randomized participants to group A, where they got the probiotic for 3 weeks, washout for 5 weeks, and then the placebo for 3 weeks, and the other group got the opposite. The study started in late summer and finished in late fall. We ran pretty much everyone at the same time, but it was a long study. This is the IL-10. These are the IL-10 data that I was telling you about in the allergy study. So let me walk you through this. This is IL-10 concentration. This was one group, they started on probiotic. This was baseline. They got the probiotic for 3 weeks. We measured their IL-10 again here. This was the 5-week washout period, and then they got the placebo. This is their baseline and final. If you notice here, the probiotic resulted in an increase in IL-10 concentration, but the IL-10 didn't go back down to normal, down to this level. We had a carryover effect, so it remained high. Okay, so now it's starting from here with the placebo, and we saw an increase in IL-10 with the placebo here and a bigger increase with the probiotic. That is a period effect. So what we think happened was, this was a long study. People eat different in summer than they do in fall. People get influenza or flu shots. They get exposed to cold and flu. So there's that cough. So we think that never came down because, excuse me, because of the environment that they were in, making it very difficult to interpret. The other thing you can do, I'm so sorry. Okay, the other thing you could do is run a quality study. Make sure all of your study coordinators are trained. Standardize your methods. I'm so sorry. Try one more time. Standardize your methods, use standard operating procedures. We run all of our participants pretty much at the same time. I need a large study crew. We all need to be saying the same thing. So we train, we role play every scenario, and we have standard operating procedures. You can't have a lot of deviations of protocol. You have to follow the protocol. Everyone has to follow the protocol. That'll decrease your variation and allow you to see differences, find measurable differences. The other thing you need to do is increase your sample size. You can also add covariates to the model. I mentioned add sex into your statistical analysis to partition out the effect of males and females or BMI or whatever. But again, if you start adding all these covariates, the target population can change as well. So this is the study that I did with the GOS and the cold and flu, and this represents the average percentage of days of cold and flu. When we added all these covariates in the model, we had an effect of BMI category and intervention. So there's an interaction between BMI category and intervention, where those of a healthy BMI, we saw a decrease in average, a 40% decrease in average percentage of days with cold and flu with a 5 grams of GOS. We did not see that in the higher categories. We had most of our participants were in this BMI category, and we had many fewer here. So this may not be as reliable as you can see by this huge standard error bars. But so that means that I was able to decrease the percentage of average percentage of days of cold and flu, but only in healthy weight individuals with 5 grams of GOS. So you can see how my by adding the covariates, my population that I was studying actually changed. All right, so for the second half, key takeaways, hopefully, that you got out of this is that you need to exploit times when outcomes are greater in magnitude or frequency. So if you're developing a model, you want a time when you're most likely to see a lot of symptoms or a lot of change. Then you need to match the mechanism of action with the intervention. Possibly increase the sample size or length of the study so that you include more days with that outcome that you're looking for, and then decrease study-related variation. I made it to the end without coughing anymore. So want to thank you for your time and thank all my students and my collaborators, and I'd be happy to answer any questions as long as I can still talk.
Thank you so much. We're starting to get more questions in through the chat, so I will go ahead and get started. So Dane Jacob, one of our grad students, asked how some women opt to skip the placebo pill phase of oral contraceptives. How do you look to see if they do not experience the mentioned GI symptoms?
If they skip the placebo, you mean they're not taking the hormone pills, or
not taking the sugar pills? I think not taking the placebos in their 28-day cycle pack, instead just going straight to hormone.
Well, we train our participants. I mean, they actually bring in their oral contraceptive to our lab, and we train them on, okay, this is your, it's amazing how much women don't know about themselves. Like, this is day one. Okay, this is when you're going to start menstruating, when you finish this pill and you start taking this pill. And so we go through all of that because they need to tell us on the first day they menstruate, and we kind of have an idea of when it's going to be. But you're right, women mess up all the time, and we use Qualtrics for our questionnaires, and so we are looking at them every day. And if we expect a woman to menstruate that day and she doesn't, we may contact her and say, hey, where are you? So yeah, participants, funny things.
Similar, Dane asked this question as well, and I think some of it may have to do with the, like you were starting to talk with, the tap dance between drug treatment and supplement. But his question was, have you done or seen any work in individuals undergoing chemotherapy, particularly, or any other sort of treatment that would have rapid GI turnover with respect to pre or probiotics are helpful? And if so, which strains? And I'm gonna be quiet, Dane, because I know some of these answers for research, but.
This question just came out of the blue.
I don't know, it's this question. Yeah.
I'm undergoing chemotherapy right now. for breast cancer. And I did that research. I am actually taking tributyrin. I've gone right to the short chain fatty acid. So I'm taking that and taking inulin because I want to maximize my short chain fatty acid production. I've switched my diet closer to a Mediterranean diet so I can have more fruits and vegetables and increase my short chain fatty acids. I did look up some of the probiotics, and I am taking Dan Active, and I'm looking for LKCI are the ones that when I read the label, that's kind of what I try to grab. So that's what I'm doing after I did personal research. So Jennifer, what would you suggest? Is that?
That's exactly what I've seen when we talk about pre and probiotics and where it's useful, particularly in, well, again, it's for medical nutrition therapy. It's part of supporting disease treatment. So yeah, those strain specific ones are so important, and the exact same ones that I've read as well. Yes.
Very cool.
Yes. And good luck in your fight, and keep your chin up. I'm sure that you'll definitely, we say it's a ton of feathers still weighs a ton, but all of those feathers with all of these impacts, I'm sure will have great effects. Dr. Parks, did you want to ask your question since you're next in the queue?
We have seminars all the time, and it's so great to hear somebody talk about how to maintain quality because the human studies are so hard to do. So thank you for that. And then my question, and I think about this a lot, is how much do you control? You're controlling exercise or background diet or whatever. Doesn't that choice of control depend on if you're early in your research and you're trying to find out, is there even an effect? And so you want really narrow variability. And then later, when you're trying to ramp up and look at generalization, don't you like reduce your eligibility criteria there? Or how do you think about it?
I guess I go for the kill. I go for where I'm going to find the biggest response because I just love piecing the models together and trying to maximize the response. Also, it's very difficult to publish data that doesn't have a big effect. But I will say that my statistician was amazing, and she could find statistical significance, but the effect size was probably not all that fabulous. And so now there's a lot more emphasis on effect size, and you need to report your effect size. And so what that means is, like in my student study, let's say I had a decrease of one day or whatever. So that's how many people taking it, and it just means you have 1 day less over 3 months. Well, that's not a very big effect size. I mean, I'd rather spend my money on a probiotic that protected me for months, and it may just do 1 day. So I would say now my research would change a little bit because I have to report effect size and think about that. But early in my career, I was going for fun, to be honest with you.
I actually think also what I'm hearing you say is you're a physiologist, right? So you're looking at mechanism and you want to optimize your study design to see, does the body work this way? As opposed to a public health person who's trying to really look at how does the treatment affect a population? So maybe it's not the pathway of research, but maybe it's your curiosity and the kinds of questions.
Yeah. To follow up on that, I had a question in line with the same topic. So your study where subjects were supplemented with 0, 2.5 and 5 grams of galacto-oligosaccharides, and there was a reduction in GI symptoms, I think. I saw that the differences, the changes were so little. So I was just wondering how clinically meaningful is a reduction in GI symptom of say just 0.2. I don't know what the rating scale that you use, but the changes were so little, and I was wondering the clinical relevance or how important those small changes were. They were statistically significant because you were powered, you had large sample sizes. Yeah.
But the effect size was very small. So the scale went from 1 to 7. And you're right, we saw just like a point something difference because we had a huge sample size. So if you have a big sample size, you can find a small difference, which might be biologically meaningless.
Yeah.
But if I were going to go over exams, I always knew that I had GI issues. I probably would take the gauze,
Muted.
Sorry, this is what I was talking about. So she's trying to figure out how biology is working, and you've asked a clinically relevant question, right? It's just a different way of looking at stuff.
Yeah.
We always get, no matter how basic science we are, we always get the clinically relevant question, because that's what people want to know. And so sometimes it's literally where you're coming in on the science. And I guess I need to make it clear that I might be testing something and I'm not in the clinic. I'm over here in more basic physiology. And so then I would control for everything because I want to see an effect. Yeah.
Right. And you want to be able to trace the mechanism too.
Right.
Yeah. Similar to that, with thinking about mechanism, Dr. Vera Potter asked specifically, given the hormonal relationships, any thoughts on specific benefits of soy phytoestrogens on gut microbiota?
Wow, that's a good one. I'm not sure I've seen a lot in that area, and maybe it's just my bias because I work more with the prebiotic, probiotic, and actually fibers. So I don't know a lot about the soy, but I know that I'm trying to do a study now looking at phenolic compounds, so non-carbohydrate compounds, and that's challenging too, to find differences. So I don't think phenolic compounds will be classified as prebiotics anytime soon. I think everyone's going to have a hard time finding health outcomes. Although I have to say that the definition of prebiotic doesn't just apply to the GI tract anymore. It could be to urinary tract infections, the bladder, it could be due in the vagina, etc. So maybe some of those other things will have an effect in other mucosal surfaces.
Thank
Absolutely.
you.
Yeah.
Certainly lots of space in this area. Great work. Thank you.
But those were all from the chat. So I wanted to leave some space for others if I'm switching my view though, so that I can see the gallery in case anybody has their hand raised.
I have it's more of a comment than a question. I know how that sounds, like a lot of memes about that. But I really appreciated you pointing that the inclusion versus the exclusion criteria out, and also talking about how NIH is focused more on broadening, inclusion as opposed to narrowing it down. But I feel like with this increased push towards precision nutrition now, they might be open to focusing only on specific groups at one time, because we're not going to be able to do precision nutrition studies unless we really focus the studies on select individuals or specific groups of population. So I feel like maybe it's not apparent right now, but I think in the near future, I do see this move happening. So I think your work it's just going to be just a fantastic, and it has so much potential with the NIH as well.
A very good point about the N of 1 studies. I agree. They're going to have to change the way they think.
I think the rule is to leave 6 seconds of silence, right? To allow for questions. So. And 6 seconds always seems so long when you're counting in your head or when you're trying to wash your hands, correct? Thank you so much for joining us today. I've really enjoyed today's talk. It is refreshing to see science, nutrition science from two sides, two very relevant sides, from the food side and the physiology side, which I really appreciated. So I'm taking a cue from Dr. Parks. For anyone that can unmute and physically clap, we'd love that. If you can use your emotion reactions there for clapping, we'd appreciate it. But thank you so much for your time today, and we really enjoyed your talk.
Thank
Thank
you.
you.
It's great.
Thank you.
Bye-bye.
Bye.